Hello, everybody. My name is Luke Leman. I'm an assistant professor of chemistry at Scripps Research in California. This is the first of a two-part talk along with Christine Keating covering processes acting on building blocks, assembly, and complexification. I've decided to focus my presentation on polymerization reactions of life's building blocks. So my subtitle, I guess, is How on Earth or Elsewhere Did the First Polymers Form? This talk is aimed at a very diverse audience, so I've done my best to focus on the key concepts rather than get bogged down in the details. And I've tried to provide key citations throughout the talk so you can go have a look at specific papers if you'd like to follow up on the details. So why focus on polymerization? Polymers have incredible emergent properties. The polymers of life provide structure, compartmentalization, catalysis, act as motors, they store and transduce information, and on and on. So I think it's fair to say that the most important molecules in known biochemistry are polymers. And by extension, polymers of some sort probably played a critical role in the transition from non-living matter to life. The earliest polymers were not necessarily the same as those that biology uses today, and were probably more heterogeneous due to the presumed messiness of prebiotic reactions. But that said, polymers were probably required for the transition from prebiotic chemistry to life. What makes polymers special? Why are they required for life as we know it? There are several reasons. First, polymers allow easy access to huge sequence space. What I mean by that is a huge diversity of different sequences can be produced from just a limited number of monomers. So 4 nucleotides for nucleic acids and 20 amino acids for proteins are used to specify the whole of the diversity in the tree of life. Also, the use of polymers provides biology with, in some sense, an energy-saving economy for synthesis and recycling. The same biocatalysts and building blocks can be used, recycled, and reused to synthesize many different molecules. Another crucial feature that makes polymers critical for life is that polymerization promotes folding. Linking together monomers into a chain forces those monomers into close proximity and dramatically reduces their degrees of freedom. So the presence of a backbone imposes interactions between monomers, which in some sense starts the ball rolling down the energetic funnel of folding and assembly. This is true for all the classes of biopolymers. Free amino acids or free nucleotides, with very rare exceptions, do not associate with each other. So free G and C don't form base pairs in solution, whereas in the context of an RNA, they obviously form hybridized duplexes. Proteins form folded globular structures with hydrophobic cores, but monomeric amino acids don't associate and don't assemble. And the same is true for polysaccharides. Polymerized sugars assemble into stable fibers and make cotton that we're wearing in our clothes, but monomeric amino acids don't do that. So polymerization fundamentally changes the properties of the constituent building blocks and enables the formation of remarkable folded structures. So how did the first abiotic polymers get here? I think it's instructive to first briefly consider how biology synthesizes these polymers before getting into the abiotic syntheses. Proteins, nucleic acids, and polysaccharides are universally formed via condensation reactions, which release water to join together their monomers. Proteins are synthesized by condensation of amino acids, nucleic acids from condensation of nucleotides, and oligosaccharides via condensation of monomeric sugars. This poses a major problem for prebiotic chemistry because condensation reactions are thermodynamically unfavorable in water. So in current biology, these reactions are catalyzed by enzymes and energetically driven by pyrophosphate hydrolysis. But of course, biocatalysts and metabolism to produce energy-rich phosphate species wouldn't have been available for prebiotic reactions. So this is the conundrum. All of the biopolymers are synthesized by dehydration reactions that are thermodynamically unfavorable in water, which is the solvent of life. They do not occur spontaneously to any significant extent. So how could polymers have been synthesized via condensation reactions before the evolution of biocatalysts and metabolism? A number of proposals to overcome this difficulty has been made, and the proposed polymerization reactions can be divided generally into heating melting processes, hydration dehydration cycles, and chemical activation of the building blocks. The conditions for these reactions vary widely in terms of pH and temperature and metal ions present. And I want to point out that these reaction driving forces are not mutually exclusive and certainly could have complemented each other and been occurring at the same time. Another important thing to keep in mind is the dynamics of polymer formation and breakdown that would have been at play. It seems likely that there would have been a continuous generation and breakdown of protopolymers due to dehydration reactions, forming them, and spontaneous decay of the resulting polymers via hydrolysis, leading to something like a steady state. This dynamic balance over a large number of cycles, where some oligomers were more stable and some were less stable, could provide a mechanism for enrichment of certain oligomers. So now I'd like to briefly go through some examples of each of these driving forces I've mentioned and mention their scope and limitations. Okay, heating of building blocks is conceptually the simplest way to provoke them into oligomers. Heating and wet-dry cycling processes have been reported for each of the polymer classes. On this slide, I'm showing an example of wet-dry cycling for glycine to form peptides and wet-dry cycling of glucose to form oligosaccharides. Repeated cycles of heating and cooling amino acids have been done at a variety of temperatures and under a variety of conditions. Generally, these reactions produce peptides, albeit in low yields and relatively short lengths. Similar reactions for oligomerizing nucleotides have been carried out. But again, to summarize a lot of work, heating of the building blocks can lead to short oligomers, but these reactions are generally complicated by the formation of side products, poor regioselectivity, and varying degrees of degradation. Similar results have been found in model hydrothermal vent conditions, both for nucleotides and peptides. Dimers and trimers of adenosine monophosphate were obtained in environments simulating hot vents on the seafloor. Similarly, with simulated hydrothermal vent reactions for glycine, diglycine and triglycine were formed in low yields along with diketopiperazine. In general, these processes on their own only produce short oligomers in low yields. And the limitations of these I've listed on this slide. These reactions can give rise to complex branch structures. There's a low threshold between forming product oligomers and their decomposition. And then more generally, in a dry state, there could be issues with poor diffusion, potentially long exposure to damaging radiation, and drying processes are potentially harmful to life forms that may have arisen due to disruption of membranes or preventing access to food or energy sources. Okay, so now moving on to condensing agents. The results for condensing agents, honestly, are not much more encouraging. The idea is that certain reagents can couple the free energy of hydrolysis of the condensing agent to drive a polymerization reaction uphill. This slide shows a timeline of various potentially prebiotic condensing agents that have been reported for amino acids. On the x-axis is the yield of peptide for the most efficient condition reported. And the y-axis is the year. As a whole, these reagents are not very satisfying. Only trimetaphosphate and carbonyl sulfide give yields better than 25%. And even for those reagents, there are questions about their plausibility and abundance as prebiotic molecules. These condensing agents also often lead to the formation of side products. Likewise, for nucleotides, attempts have been made to polymerize monomeric nucleotides in aqueous solution using agents like cyanamide or water-soluble carbodiamides. But again, these efforts typically result in the formation of only dimers to tetramers in low yields. Polymerization of nucleotides have most often been studied with pre-activated building blocks. And the most commonly used of those are phosphorimidazoles, shown here on the top left. The cyclic phosphates, cyclic 2,3 cyclic nucleotide or 5,3 cyclic nucleotide monophosphates have also been studied for polymerization. In the absence of a template molecule, these activated nucleotides will oligomerize, but only poorly, and generally produce short, linear, and cyclic oligomers. Also, there's an issue of regioselectivity. Typically, these reactions, in the absence of a template, give a mixture of the 5' 3' and 5' 2' phosphodiester linkages. These reactions can be promoted somewhat in the presence of certain metal ions. Okay, so earlier I mentioned some of the limitations of the thermal processes. Likewise, I want to point out some limitations for condensing agents. First, they don't generally yield long polymers when used in situ. And by that I mean in a one-pot reaction where unactivated amino acids or unactivated nucleotides are used. Typically only short oligomers are formed. Longer polymers can be produced when pre-activated nucleotides like the phosphorimidazoles or pre-activated amino acids like N-carboxy anhydrides are used. But the prebiotic plausibility of starting with those pre-activated reagents is questionable. Second, condensing agents often give rise to side products. So for an example, if you look at the HPLC trace on the top right of this slide, you can see evidence of both only short oligomers and side products being formed. In this case, hydantoin and urea side products. Third, condensing agents could only have been effective if they were continuously supplied in a given location. So the continual hydrolysis of polymers would mean that polymers could only build up if a condensing agent was supplied continuously. And this limitation has been treated theoretically by this paper from Ross and Deemer. So far everything I've told you is in general disappointing in terms of generating abiotic polymers. We can produce oligomers, but generally only short ones and not in very good yields. So what are the problems? I would say it's complicated and highly dependent on reaction conditions, but there are some generalizations that can be made. First, side reactions often limit these reactions. Poor solubility of products can be an issue. Polymer hydrolysis will always be a problem as long as these reactions are occurring in the presence of water. And lack of reactant organization. What do I mean by that? By that I mean low concentrations of reactants and poor pre-organization for polymerization. So luckily for us, there are some ways around these limitations that have been demonstrated. Certain mechanisms can minimize or even take advantage of cyclization. So for example, in polymerizations that produce polyesters, those reactions are thought to involve the participation of a cyclic dimer species called morpholine diones as activated intermediates. So in this case, cyclization of an intermediate produces a more reactive intermediate that rather than a dead end product. Solubility can be overcome by using monomers with polar side chains or alternative solvents. Polymer hydrolysis can be overcome in environments with low water activity or again with alternative solvents. But the issue I would like to focus on in more detail is that of reactant organization. So I'm going to share a few examples where polymerization reactions are promoted using either surfaces or interfaces or template molecules and even self-assembly. Okay, so as a first example, oligomerization of nucleic acids and peptides both can be promoted by mineral surfaces. And the mineral that's been most studied here is montmorillonite. Ferris and coworkers have studied oligomerizing nucleoside phosphorimidazoids and related activated nucleotides for many years. And montmorillonite was found to catalyze the formation of RNAs up to 50 mers in length. These products had mixed 2' 5' and 3' 5' phosphodiester linked bonds, although with preference for the biological 3' 5' linkage. The regiospecificity of this reaction has been proposed to result from an increased local concentration and organization of monomers that are absorbed to the surface in the mineral's interlayer between the clay plates, although the exact mechanism is still not well understood. As a second example, abiotic oligomerizations can be promoted at interfaces. Examples of interfaces that have been reported for this are the surface of a lipid vesicle, in between lipid bilayers in a dried multilamellar vesicle, or at the air-water interface. Luisi and colleagues found that certain amino acids would selectively partition onto the surface of lipid vesicles, and that this partitioning would increase their local concentration and thereby promote oligomerization. And as you can see in the graph on the left here, the obtained peptide lengths were much longer with liposomes compared to the same condition without liposomes present. In a somewhat similar sense, Deemer and coworkers have studied polymerization of nucleotides that occurs when they are dried in the presence of phospholipids, and have found that multilamellar vesicles can promote the formation of what they call RNA-like polymers. So along the same lines as for montmorillonite and the vesicles in solution, it's proposed that the nucleotides are concentrated and organized within the lipid matrix, and that the condensation reaction is favored due to the absence of water and the elevated temperatures of the reaction. And X-ray scattering studies have shown that rather than forming a random array, the AMP molecules are organized in a regular array within these multilamellar dried states, with the phosphate and ribose groups in close proximity that can facilitate polymerization. Okay, template-directed synthesis has been extensively studied in the context of nucleic acid chemistry, and to a lesser extent for certain peptides, such as ones that can form amyloid-like fibers. Template-directed synthesis is a process where oligomerization is promoted by reactant pre-organization along a template molecule. Templating reactions of this sort can show good fidelity and can be fairly selective for chirality and regioselectivity. The basic idea is that incoming monomers can recognize the complementary position of the template, and monomers that are able to hydrogen bond or form an attractive electrostatic interaction or otherwise chemically pair with the complementary position of the template will have a longer lifetime at that position and thus a higher likelihood of being incorporated into the growing polymer. Okay, I'm only going to briefly mention fragment ligations, but suffice it to say that another mechanism for building up larger polymers is by ligating together shorter fragments. This can work with the caveat that the shorter fragments need to come from somewhere, so there still must be some abiotic oligomerization to produce those fragments. This slide shows an example of a reaction where 7 shorter fragments are ligated together with diamidophosphate, which is a potentially prebiotic condensing agent, over a period of a few weeks. These reactions were carried out in the eutectic phase of a water-ice mixture, which Diemer and colleagues earlier had showed that freezing solutions of activated nucleotides could substantially promote their polymerization by essentially excluding the activated nucleotides from the ice lattice and thereby concentrating the reactants. So in some sense, this is another example of reactant pre-organization. Okay. One issue that is not often dealt with experimentally is heterogeneity. What I mean by that is very often a pure monomer is used for studying polymerization reactions. Now, of course, this is not at all prebiotically plausible, but because there would have been many different building blocks present simultaneously on the early Earth. So some sources of heterogeneity are shown here. On the monomer side, that can be from different stereochemistry in the building blocks, different side chains in the building blocks, and closely related monomers that can react in similar ways but would result in the production of different molecules. On the polymer side of things, there's the issue of regiochemistry, where monomers that have 2 or more of the same type of functional group can give rise to different products depending on which of those functional groups reacts. I've shown this here for the example of a ribonucleotide, which can react at one of 2 hydroxyl positions, either the 2' or the 3'. And depending on which of those positions reacts, the resulting molecules are different. Heterogeneity is often considered to be a problem in the context of prebiotic chemistry because life's biopolymers are homogeneous. Proteins are made only of L amino acids. RNAs are made only of 3', 5' linked nucleotides. But there's been a growing appreciation that heterogeneity can't be ignored and in some cases can even lead to interesting chemistry, and that could facilitate prebiotic chemical evolution. One example of this comes out of work looking at abiotic oligomerizations of alpha hydroxy acids to form polyesters. Hydroxy acids are closely related to amino acids and are found together with amino acids in meteorites and in model prebiotic reactions. And it turns out that alpha hydroxy acids polymerize more readily than amino acids, so meaning at lower temperatures and with better yields. And it has been found that if amino acids and hydroxy acids are mixed together and polymerized, the hydroxy acid will promote the formation of peptides through a process called ester-amide exchange. So here, including 2 different types of monomers, amino acids and hydroxy acids, actually makes the formation of peptides more efficient and able to proceed under milder conditions. Thinking about backbone heterogeneity and the consequences of that for prebiotic reactions raises the issue of selective or preferential reactivity. The idea here is that certain building blocks may react differently or selectively relative to other ones. And so even if the pool of reactants is large, selective reactivity can produce sets of molecules that are enriched in certain building blocks compared to others. So I'm going to highlight 2 examples of this, although this is an active area of research with many interesting examples. One of the long-standing questions in origins of life research has been how the proteinaceous side chains were selected. And Frankel-Pinter and colleagues studied the oligomerization reactions of a group of positively charged amino acids shown here on this slide in hydroxy acid catalyzed dry down conditions. And they observed that these proteinaceous amino acids oligomerize more extensively and with greater selectivity at their alpha amine compared to the non-proteinaceous amino acids that were examined. These findings provide a chemical basis that could have contributed to the selection of certain amino acids for incorporation in the set of amino acids used in proteins. So as a second example of selective oligomerization, in polymerizations of N-carboxy anhydrides, which are pre-activated amino acids, it turns out that the incoming monomers are selectively incorporated to match the chirality of the growing chain, leading to overall stretches of predominantly homochiral products. So in short chain, the incorporation of the L versus D enantiomer is random, but as the chain grows longer and longer, there's a greater influence of the chirality of the oligomer on the incoming N-carboxy anhydride. And that results in an increasing homochiral excess as the oligomer elongates. Okay, so where does this leave us? Stepping back for a minute, there are several different ways to produce or promote the coupling of building blocks abiotically into oligomers. And it's quite satisfying that there are a number of different mechanisms because that makes it more likely that at least some of these processes could have been occurring in different environments on the early Earth. But, and this is a big limitation, none of these processes can plausibly make polymers even close to the lengths that compare with those typically at play in biochemistry today. Proteins are on average range between 200 to 400 amino acids in length, which is much longer than any known prebiotic process can achieve. And the same is true for nucleic acids. The range of lengths of RNA used in biology are from 50 to 100 nucleotides and longer. And this is longer than can typically be obtained in known prebiotic processes. So there's a large gap between molecules that can be made abiotically and the molecules that biology uses. Now, I want to be clear that I'm not suggesting the origin of life required polymers this long, and it's dangerous to project too much from biology today back to the origin of life. But I also think this is an important gap to point out so that nobody is under the impression that this problem of abiotic polymerization is solved. It's possible that we just haven't found the right conditions or chemistry to allow the synthesis of long abiotic polymers. But on the other hand, long polymers probably weren't necessary for the origin of life. It seems likely that the earliest life made do with shorter oligomers and relied on non-covalent assembly of these molecules to bootstrap the way to longer, more sophisticated polymers. So consider what we know from today's biochemistry. Biological polymers are nearly always associated with other polymers. Assembly is ubiquitous. Around half of all proteins exist as multimers. DNA molecules assemble with their complementary strands, and RNA molecules extensively associate with proteins and other RNA. So forming molecular assemblies with other polymers is almost the default state for proteins and DNA and RNA. And there's every reason to believe that the earliest oligomers would likewise have self-assembled and associated with other types of oligomers. So while it's true that abiotic processes don't produce oligomers as long as those we find in biochemistry, that probably wasn't necessary because short oligomers that can assemble were probably enough to get chemical evolution started. I just want to give a couple examples that highlight this. Okay, so first, short RNA fragments can self-assemble non-covalently to produce active ribozymes. This means that a given ribozyme does not necessarily need to be a single long polymer. Instead, shorter oligomers can come together noncovalently to create a larger functional unit, as has been demonstrated in this paper. And here's another example. David Lin and colleagues showed that 7-mer peptides could self-assemble into amyloid-like long fibrous structures that position functional groups appropriately, such that these supramolecular structures could catalyze a retroaldol reaction. So whereas biology has evolved long protein enzymes that fold up into precise three-dimensional shapes to catalyze reactions, prebiotic oligomers could have, in some sense, achieved the same outcome, not by using long polymers, but instead by using self-assembly of much shorter oligomers. Okay, so to summarize, the take-home messages from my presentation are listed here. The tapestry of biochemistry is woven with polymers. Polymers are among the most important molecules in known biochemistry, and by extension, polymers of some sort must have played a role in the transition from prebiotic chemistry to life. The leading ideas for driving forces to promote abiotic polymerizations are heating or melting, dehydration, hydration cycles, and chemical activation of the building blocks. And I talked about some ways of promoting polymerization, largely based on pre-organizing the building blocks on surfaces or on interfaces or on template molecules. A major unresolved challenge in the field is to more fully explore the consequences of heterogeneous monomer mixtures and the products that would result from them. Another major issue is that oligomers produced in known abiotic processes are much shorter than polymers found in today's biochemistry. And I talked about one possible way around this problem, that being assembly of oligomers into non-covalent complexes in ways that could have aided in bootstrapping up function from relatively short oligomers.